Hi Year 11, this is a quick exam review of the B1 Foundation specimen paper that you would have taken at the start of April. What you need to make sure you're doing as we go through it, as I've said to you in lessons, make sure you've got a green pen ready and you're going through and correcting any answers that you got wrong, but also you're making a note of the revision topics that you feel that you need to put a little bit more effort into at home. Okay. If you've got any questions on any of the answers that we're providing throughout the video, please make a note of them and then come and see me at some point throughout the week when you're in school, folks. Okay. Right, question 1. So the diagram shows how pigs can be cloned. We know straight away from that it's a cloning question. So it shows here your adult male, the adult female. We've got a cell that gets taken away from it. We've got another cell here, D, that gets removed and something else is removed from that cell. It takes an empty cell. They come together, they make an embryo, and then eventually that forms into a cloned animal there, so it gets implanted into a surrogate. So the first question, 1A part 1, which structure A, B, C or D is an egg cell? Well, your egg cell is your female sex cell or your female gamete, therefore the egg cell is going to be D because that is taken from the adult female. Okay, so that one should be D. Next one, the nucleus. So we can see that the nucleus of the egg cell has been removed. So the nucleus is going to be C. And an embryo is a bundle of cells after the first division when a sperm and egg come together. However, we've not had a sperm and egg come together for this one. We've just had this cell resulting here. And the embryo is going to be this bundle of cells here at B. So the answer for that one is B. Nice easy question to get you started on. 1B, walking onion plants grow a bunch of bulblets, which are tiny bulbs. The bulblets start to grow and the stalks bend over with the weight of the new growth. This makes the onion plant seem to walk across the garden. So we've got a labelled diagram here, which describes exactly what we were talking about just. Producing plants in this way is called asexual reproduction. Use words from the box to complete the following sentences. So we have got asexual reproduction needs only one. Answer for that one is parent. So asexual is one parent. Sexual reproduction always involves two parents. Asexual reproduction does not involve production of a gamete. The gametes are the sex cells, so for example the sperm or the egg, or the pollen in the egg in plants. But in this example, if we're doing asexual reproduction, that involves none of those. Okay, and the daughter plant cell is called a clone. The reason for that is because it's genetically the same as the adult plant that was in question, so it's going to be exactly the same as the starting plant. Okay, moving on. Question 2. The diagram shows how the number of species in different vertebrate groups change between 400 million years ago and 5 million years ago. The wider a block is, the more species there are. So we can see, as we look down here, that some of the blocks get wider, and it says the wider it is, the more species there are. This is important for one of the first questions. So make sure, please, that you are reading all of the question before you attempt to answer it. Which group had the most species 200 million years ago? So if we look along 200 million years, you're looking for the branch which has got the widest point on it. And as you can see, it's this one here, which is the amphibians. So our answer for that first one is amphibians. Okay, question 2B. To which group are birds most closely related? Well, we can use the evolutionary tree here and trace it back to the nearest branch where it joins to something else, and the branch that it joins onto is the reptiles, which is this bit here. A couple of people were saying mammals. You're not looking next to the bird group, you're following it back and seeing which one it connects to. So that one should have been reptiles. Okay. And then 2C, complete the following sentence. A study of fossils gives evidence for the theory of evolution. Reason for that is we're seeing what the animals used to look like in the past, and we can see how they've changed over time compared to animals today. So it's 1 mark for each of those. Question 3. Gardeners often collect fallen leaves in autumn and place them on compost heaps. Over the next year, the leaves decay. Which living things cause leaves to decay? Range of answers you could have had for this one. You could have had microbes or microorganisms. They may have allowed it if you said decomposers. So that'd be things like bacteria. Or you could have said detritivores, which is your keyword there for small animals that feed off the remains of dead things. So from that, those are the answers that you could have had. Ones that I saw a few times but we couldn't accept were things like worms or beetles. You had to use the keyword detritivores if you were talking about those. Okay, next one, 3B. The leaves decay more quickly in summer than in winter. Give one reason why. Basic reason, one of the things that we need for decay is for a warm environment, and it's warmer in summer. So it decays faster in summer because it's warmer. And the compost heap has holes in its sides to allow gases to enter. Which gas is needed for decay? Well, those organisms that are causing decay to happen, so like your detritivores, your decomposers, your bacteria, your microorganisms, those all need to respire in order to be living, for the decay process to happen. And the gas that's needed for them to respire and allow decay to take place is oxygen, not carbon dioxide, which is what a couple of people put. Question 4. Students investigated how well antibacterial mouthwashes worked. They tested 4 mouthwashes, PQ-RNS. They spread bacteria on a nutrient jelly in a petri dish. They soaked identical discs of filter paper in mouthwashes PQ-RNS. They placed the discs on the growing bacteria. They covered the petri dish and they incubated it for 2 days. So they're placing those different antibacterial discs onto different areas of the bacteria. It says the nutrient jelly was heated to 120 degrees Celsius before being poured into the dish. Why is this necessary? It's to kill any microorganisms that may already be there. Okay, so we just want to be investigating the bacteria that we are putting on there, nothing that is already there in the first place. Okay. What is the maximum temperature at which bacteria should be incubated in a school lab? Maximum temperature in schools is 25 degrees Celsius. It can go up to around about 35 or 37. However, it's a little bit dangerous if that starts to happen because the bacteria grow too fast. 4C. Diagram 2 shows the appearance of the petri dish after 2 days. Which mouthwash, PQ-RNS, kills the most bacteria? Well, if we analyse the areas, we can see that the disc S has cleared the most area of any bacteria that may have been present. Disc R has cleared the least area, P the second least, and Q the third. Okay, so S was our correct answer because it clears the most bacteria. Okay, next question. Question 5. The photograph shows a bird called the korhan. Korhans live in South Africa. Scientists have studied changes in the number of korhans since 1997. They asked volunteers, drivers to record the number of korhans they see for every 100 kilometres they drive. The bar chart shows changes between 1997 and the end of 2008. Okay, so we can see general pattern here. We'll look at that in a second on the next page. Okay, so before we look at the graph, we've got question 5a. This method of counting korhans could have led to an inaccurate estimate of the number of korhans. Explain how. Well, in the question it says that they were driving along a certain stretch of road. Therefore, they're only going to see a certain amount of them in that particular area. So the actual number is probably going to be a lot higher than the estimate. So you've got 1 mark saying the actual number may be higher. Than the estimate. And the reason for that is because not all birds are visible. Okay. Okay. That was the first part. 5b, which statement best describes the changes in the number of korhans between 1997 and 2008. So we've got three options. There was a steady fall. The number of core hands went up and down, but there was an overall fall in numbers, and the overall number of core hands went up and down, and there was no overall trend. So if we look at the graph, we can see that we start off with our data in 1997. We see roughly just over 2 core hands for 100 kilometres of driving, and then over the years the numbers go down, but then they start to come back up and then go down again. Therefore, the one that most fits that box there is going to be number of core hands went up and down, which it does, but there was an overall fall in numbers. The overall fall in numbers is because it's ended up finishing a lot lower than what it started as. Okay, 5C. Core hands live only amongst tall vegetation in areas of the country where there are few people, which is the most likely explanation for the change in numbers of core hands between 1997 and 2008. So we've got many core hands being killed by cars. It's not likely going to be that because they live in areas of tall vegetation, so areas where there's lots of trees. Many core hands have been killed by people for food. Not really. There's no evidence to suggest that. The only one that sounds sensible is the habitat of core hands is disappearing. That could be due to something like deforestation. Okay, so with these sorts of questions where you've got to tick the boxes, please make sure that it's only one box that you tick when it asks you to. So make sure you're reading these bits here, how many boxes it's asking you to tick. If it asks you to tick 2, please make sure you do, because it'll be worth 2 marks. Okay, next question. 6. Lichens are pollution indicators. Complete the following sentence. Lichens are indicators of the gas, and that is sulfur dioxide. So the lichens can detect that pollution in the environment. The chart shows how much pollution different lichens can tolerate. So it shows you a little bit of data there. So we've got Pamelia, Dagelia, not been able to stand a lot of it. Bryoria and Fusonia can stand a bit more. Ramalina, Lecanora can stand a bit more as well, and Diploicia and Xanthoria can withstand a high level of pollution. So it says the diagram shows the areas J, K, L and M in which different lichen species grew around a factory, and the factory burns coal. So within the coal there will be sulphur, and that sulphur is going to react with oxygen when the coal burns, which is going to form sulphur dioxide. It's asking in which direction does the wind blow the pollution from the factory. Okay, so we've got the factory, which is based here, and we can see that most of the pollution, or the indicator species for pollution, have gone towards the east. Therefore, the correct answer for that is from the factory towards the east. No trick questions there. You just got to look at the diagram, understand what it's saying. And then which row in the table shows a correct distribution of lichens? Well, we know that the area which is going to have the most pollution on it is going to be the area that is arguably closer to the factory. Therefore, the ones that we're going to see, we'll bring the table back in now so we can just have another look at the data. So, like we said, the idea is that as the pollution gets further away from the factory, so going sort of in this direction, there's going to be less pollution. So J is going to contain the most pollution, K will then contain the next highest amount, L will contain less, and M will contain the least. So if we look at the diagram and then look at our options, one of the species that should be close to the factory is either going to be the Xanthoria or the Diploicia. So we're looking for a lichen in area J to be either the Xanthoria or the Diploicia. So immediately we can rule out this row here, because neither the Xanthoria or the Diploicia are in area J. In area K, we're looking for lichens that can withstand a moderate amount of pollution, so looking for lichens that are going to be about here. So that's Lecanora and Ramalina. And the only one which has got Lecanora in that area is the bottom row, which is this one. Therefore we know it's not going to be the top row, which gives us our answer, so there should be a tick in the bottom row for that one. Okay, moving on. Emperor penguins have adaptations that help them to survive in very cold Antarctic conditions, and it shows you there it basically gives you the adaptations in the question, in the diagram. It shows you that it's got oily feathers, a streamlined body, and a layer of fat under the skin. So it's giving you 3 adaptations there straight away. Some of the information from the question: emperor penguins catch fish in the sea, so they have to get wet in order to catch the fish. Use the information and information from the drawing to explain how the emperor penguin is adapted to survive in the Antarctic. So we need 3 points because it's a free marker. So our first thing that we can say is it has a streamlined body to help it swim. Okay, so that would be 1 mark. Next thing you could say, it has a layer of fat, and that will help to insulate it. So it insulates the penguin and keeps it warm. Okay, and then the 3rd mark is saying that the oily feathers are waterproof, which means when it goes into the ocean and then it gets out, the water is not going to stay on it for long, it's just going to run off it straight away. That's 3 points there that you could have included. Next one, 8A part 1. The total volume of water lost by the man was 3,000 centimetres cubed. How much water was lost through his skin? Well, what we need to do here is add up the amounts of the ones that we can see. So we've got 2,000 being lost in urine, 300 is being breathed out, and then 100 is lost in feces. So if we add those up, we've got 2,000 plus 300 plus 100, which gives us 2,400. We know that the total volume of water lost is 3,000, therefore 3,000 take away 2,400, it's going to leave us with 600. So your answer was 600 centimetres cubed. Next one. The same man lost 1,200 centimetres cubed of water through the skin on a warm day. Give 1 reason for the different volumes of water lost on 2 days. Okay, so most obvious reason, if it's a warmer day, then the person is going to have to cool down, therefore they're going to sweat more. So, person will sweat more on a warmer day, and that's to cool down. Okay, next question. 8B, to maintain water balance in the body, the total volume of water taken in must equal the total volume of water lost. Give 2 ways this is achieved on a hot day when compared to a cold day. So 2 ways that they can equal the total volume of water lost. So what you can tip there is that the volume of water in the urine will decrease, so the urine becomes more concentrated, so that's what you would see happen if your urine is quite a dark colour instead of a light colour. Volume of water in the faeces increases. That's not going to help us to keep water inside our bodies, so that's not an answer. The volume of water taken as food or drink increases. Yep, that makes sense. If it's hotter, we're going to drink more. And the volume of water breathed out decreases. That one is incorrect. So 2 ones there are water in the urine decreases, and water taken in the body through either food or drink will increase. Okay, question 9. Dr. Semmelweis collected data about the number of deaths in 2 maternity wards in the hospital where he worked. From 1833 to 1838, there were the same number of doctors and midwives delivering babies in Ward 1 and 2. From 1839 to 1847, medical students and doctors delivered babies in Ward 1, midwives delivered babies in Ward 2. Okay. Dr. Semmelweis also noticed that doctors often came straight from examining dead bodies to the delivery wards. This is why I say it's important that you pick out the information. That there will be a key thing for later. Okay, so if we move down, use the formula death rate equals number of deaths times 1,000 divided by number of patients to calculate the death rate for Ward 2 in the years 1839 to 1847. So if we look at the number of deaths in Ward 2 during that time period, it's 691. Multiply that by 1,000 is going to equal 691,000, and then we need to divide that by the number of patients, which is 17,791. We run that through the calculator, and that gives us an answer of 38.84 deaths per thousand. So this here is your answer, and that's how you get there. Okay, so you have to use the data in the table. You've got to use these two numbers here and use the formula. If it gives you a formula, please make sure that you are using it. Question 9a part 2. Suggest a hypothesis for the difference in death rates on Ward 1 and Ward 2 in the years 1839 to 1847. Okay, so what you need to do for this one then, if we look back at some of the information we highlighted earlier in the question, we can see that the doctors who came straight, the doctors who were operating the wards came straight from examining dead bodies to the delivery ward. Those dead bodies are going to contain microorganisms on them that are going to start decaying the body, and basically those doctors are carrying bacteria between the ward where there's dead patients and the ward where the women are giving birth. So what we need to point out there is that the women in Ward 1 are infected, and that is by pathogens on the dead bodies which the doctors have carried. Okay, so you got one mark there for pointing out that the women in Ward 1 got infected at a high rate. Second mark was suggesting why that was. So the doctors were carrying the pathogens towards the women. Okay, 9b, antibiotics are now used in hospitals. What is an antibiotic and what does it do? Well, basically an antibiotic is a drug, for 1 mark. Second mark, that kills bacteria. You have to say here that it's bacteria that antibiotics kill. You can't say viruses because the viruses are not affected by antibiotics. Okay, next question. MRSA is causing problems in hospitals. Give one reason why. Main reason why MRSA is causing problems is because it's resistant to a lot of the antibiotics. So it's resistant to many antibiotics. And then D, how can the work of Semmelweis help to reduce the problem caused by MRSA? What Semmelweis showed in his investigation was that these pathogens, these bacteria or viruses or whatever, they were passed on by touch, and therefore washing hands was really, really important to prevent infection. So he showed that it was important to wash hands to prevent infections or to prevent pathogens from being passed on. Okay, question 10. So a student grew a pot in an upright plant. She then put the pot in a horizontal position and left the plant in the dark for 2 days. Diagram 3 showed the potted plant after 2 days in the dark. So key bits there from the question that you need to point out. It was left in the dark. It spent 2 days there, and it was in a horizontal position, which we can see by looking at the position in figure 2 that it was left in. So we've got to explain why the plant responded in that way, and it's for 4 marks. Okay, folks, so what we need to do here was say that when the plant was flat, so when it was horizontal, auxin collected on the bottom of the stem. So when we say the bottom of the stem, the area I mean is the lower half of the stem here. Okay. Next point that we need to make sure that we say is that this is caused by gravity. So the gravity actually pulls the auxin towards the bottom. And what that hormone does, the auxin, is very, very important. It actually causes growth in certain areas of the plant. So it just so happens that in the shoots of the plant, which is what we're looking at here, auxin stimulates growth where there is the most auxin present. Okay, so that's for your next mark. So auxin causes the bottom half of the stem to grow. And then the last point is we're saying this makes the stem grow up. Let's move that down. Okay, if you've got any questions on that one, if you're not sure on how auxin is distributed in plants or anything like that, then please come and see me, because that was one of the questions that were a little bit trickier that students sometimes tend to struggle on. So anything that you want to ask about that, come and see me and I'll help you out with it. Next one. Number 11. Many diseases are caused by viruses. Children are given vaccines to protect against viral diseases. It's difficult to kill viruses inside the body because viruses live inside cells. It's a little bit different to bacteria. The bacteria are free to go wherever they want around the body, so that's because they're about the same size as one of our cells. The viruses, however, are very, very small, and they go inside the cell. They will rewrite the cell's DNA to get our own body cells to make more of the virus, which then builds up and it kills the cell, causing the virus to burst out and then infect other cells. A vaccine contains an either inactive, dead, or weak form of the virus. So the reason for that, it helps to give our immune system the chance to practice producing antibodies against that particular pathogen. And then finally, the vaccine stimulates the white blood cells to produce, I've just said it, it makes antibodies. Don't get this mixed up with antigens, please. Antigens are found on the surface of your pathogen. So let's say this is a pathogen, an antigen is found on the surface of the pathogen, and that's what the antibodies will actually attach to. I'm sorry if that diagram's not very clear, but I'm going to draw this with my hand. So the antigens, just to be clear, are on pathogen surface. Antibodies are made by white blood cells. Okay. Move that out of the way. Okay, and then we've got 11b here, which is shown as a graph. In the 1990s, many people thought that MMR caused autism in some children. This is why the Japanese government stopped using the MMR vaccine. The graph gives information about the percentage of Japanese children who developed autism during the 1990s. So we can see this data here. What we'll do is we'll look at this graph in a bit more detail on the next page when we're looking at the questions for it. Okay, so our question says the data in the graph support the view that there is no link between the MMR vaccination and autism, and it's asking us to explain why that might be. Okay, so if we look here, it's a 4-mark question, which means that we are going to have to analyse this graph in quite a bit of detail in order to give ourselves the best chance of getting maximum marks. So we're looking to see if it supports the view that there is no link between MMR and autism. So if we look at the data, we're going to pick out a couple of things first. We'll look at the general pattern. So we can see that the key down here shows that the percentage of children vaccinated with MMR is shown by the bars, and the number of children with autism is shown by the line. So we know exactly what we've got to look for there. So if we start off in 1990, 70% of children were vaccinated with MMR, and that resulted in about 36% of them actually suffering with autism. As we go over through the time, the percentage of children that get the MMR vaccine decreases. However, we see that the children who get autism, there's no general pattern there. It goes up and it goes down, it goes back up again, it stays the same. In 1995, after there's no vaccinations being taken, there's a massive spike in the number of people who are getting autism, and there's a massive fall again, even though there's been no change in the percentage of children who are being vaccinated. So all of these things sort of point to the idea that there's no link between MMR and autism, and we'll look at how we can put that down in an answer now. I'll just leave this graph here so that we can still see it. However, the writing that I'm doing might go over it a little bit. So for your first mark, we need to make reference to the idea that in 1995 there are no children being vaccinated. So in 1995 there's no vaccinations. However, the number of children that get autism, as we can see here, I'll just do it in a different colour, goes up significantly during this period of time here. Okay, so it goes from, let's have a look at the numbers, it goes from just about 20 children per 100,000 to 80 to 40 children per 100,000. So it's quite a high increase, higher than anywhere else on the graph. So we need to say that after this point, more kids develop autism. So actually give some data from the graph there as well. Talk about those figures that we've just mentioned. It goes from 20 to 40 per 100,000. And the next thing that we can say to get some marks is that the number of children developing autism actually peaked after the MMR vaccination had stopped. So you've actually got to make a comment on that. So more kids get autism after MMR stops. And that suggests that there must be something else that causes autism. Suggests another factor causes autism. Okay, so there are 4 points there. Quite nice and easy to get the marks, at least 2 marks on that. However, those were the key things that you had to say. So just make sure you're analysing the data in as much detail as possible and talking about what trends there are in that data. Okay, question 12. Some athletes use drugs containing the steroid testosterone to improve their performance. In an investigation, scientists monitored the performance of the 18 male athletes over 6 weeks of training. 9 athletes were given testosterone and the other 9 athletes were given a placebo. And it shows the results here. Okay, so we've got our testosterone group, which is here, and the placebo group, which is there. And we're looking at the mean percentage improvements in performance when they're doing a bench press exercise. So first question then, it's saying the data would have been better presented as a bar chart, and we need to give a reason why that is. The basic answer that's on the mark scheme is that there's not really enough data for a line graph. So there's not enough data. I also accepted it if you said that the data was categoric and not continuous. So by that, we mean the data is put into categories, and the 2 categories are either the testosterone group or the placebo group. Okay. Next question, suggest what was given as a placebo in this investigation. So a placebo, one of our keywords from the topic for drugs and developing drugs, is basically a fake drug. So it's something that is given to a subject, so by subject we mean a patient, but it doesn't actually contain the drug that they think they are getting. So you could suggest that for the placebo they get an injection, but the key thing it has to mention is that there is no testosterone. Okay, next question. Describe the results of this investigation. We're going to bring in the graph from the previous page, and then we'll look at that in a bit of detail. So just pause the video. Okay, so the graph should be on the screen now, and if we look at the graph, we've just got to describe the results of the investigation. We're not going to explain why it happened, so make sure you're looking at your keywords, your command words in the question. So describing this graph, we can see that the testosterone group increases over the 6 weeks to a much higher percentage improvement than the placebo group. It's actually at about 13%, whereas the placebo group, we'll just read off for that, that's at about 3%. Okay, so there's an overall increase after the 6 weeks. of about 10% in performance. So in terms of getting the marks for this question, we have to say the testosterone group improved more than the placebo. And the reason for that, well, you just got to give a quantity, you've got to give a figure to show that. So you could say after 6 weeks, testosterone testosterone group improved by 13%. Placebo group was 3%. Okay, sorry if you're struggling to read the writing there. So yeah, testosterone group improved more than the placebo group. After 6 weeks, testosterone group had gone up by 13%, and after 6 weeks for the placebo, it went up by 3%. Okay, so that was what you need to put for that. I'm just going to leave that there. If you need to copy down any of those marks that I've just got down, make sure you do it now, because I'm going to move this little bit here away from the screen. Okay. And 12D, most internet advertisements for testosterone state that athletes need to use testosterone for at least 10 weeks to significantly improve performance. Do the results of this investigation support the statements? Answer for that is no. And that's because we saw that it had improved more than the placebo group by 10%. So it performed better than the placebo. That's just for 1 mark, so that's all that you need to say for that. Okay, last question then, question 13. One that was generally answered quite poorly, but this is now your chance to try and improve it a little bit if possible. So it's on the topic of energy transfers and energy and biomass, and looking at food chains, what happens to the energy that actually gets taken in by the animals in the food chain. So we've got a diagram showing a food web for some of the organisms that live on moorland. Only a small percentage of the sun's energy captured by the heifer is eventually incorporated into the tissues of the body fox. So what that means is that as the rabbit eats the heifer, the stoat eats the rabbit, and the fox eats the stoat, only some of the energy is going to get passed on, not all of it. It's only about 10% of the actual energy amount gets passed on. Explain as full as you can what happened to the rest of the energy captured by the heifer. And it is a 6-mark question, which means there are 6 things that we need to try and mention for this. Marks, well, the mark scheme actually says that for 1 to 2 marks, there is a brief explanation of at least 2 ways energy captured by the heifer is transferred, which has little clarity or detail. For 3 to 4 marks, there is some explanation of a range of the ways in which the energy captured by the heifer is transferred. And then 5 to 6 marks, there is a clear, balanced and detailed explanation of a large variety of ways in which energy captured by the heifer is transferred. Okay, so we've got to give as many ways as possible to get the higher marks on this. So our first point that we can say is that some of the energy will be used for respiration. So respiration actually releases energy. Okay, so that's your first one. The next one, energy can be lost as waste. Third one, it's used to repair. So if there's an injury or any damaged tissue or cells, it's used to repair those. Fourth thing you could have said was for movement. Fifth thing could be lost as heat. So we lose energy, especially if you're warm-blooded through heat. And then your final thing that you could say is that some organisms actually die, and rather than them actually being eaten, we have decomposers or detritivores which recycle with the energy. Okay, what you should not be saying in this sort of question is that, if we go back up, the heather starts with the energy, the rabbit takes some of the energy, the stoat takes some of that energy, and the fox takes some of that energy. That is not what the question is asking you to do. If you look at it carefully, explain as fully as you can what happens to the rest of the energy captured by the heather. So it's not talking about how the energy gets passed on, it's talking to the other animals in the food chain, it's talking about what actually happens to the energy that it's using. Okay, so I'll just show you those again very quickly. So respiration releases the energy, energy is lost as waste, it can be used for repair, it can be used for movement, some energy is lost as heat, and then finally when the animal or plant dies, decomposers or detritables can recycle that used energy. That's the end of the paper, folks. If you've got any questions that you want to ask, or any particular topics that you're still not sure on, or you're not sure which topics you need to revise because you did poorly on a certain question, make sure please that you come and see me, and I will give you a little bit of a push in the right direction. Okay, thank you folks.